Go. Hi, Barry.
Hi, Adam.
What do you want to talk about?
I don't know, anything, you always bring up the good point.
Well,
I've been so busy this week. It's been crazy. Absolutely. I've been working from 6 in the morning till 10 at night every day. I just, the beginning of the academic year, I think it's something about Zooms that it makes it so easy to have meetings. People are just creating more and more meetings and there's so much going on. I can hardly get any work done.
same boat, but
Anyway,
well, we were talking about a couple different things before. So we're talking about the nature of body maps in the brain and how those might differ across individuals and maybe species. So like
definitely.
you might think of like a human's homunculus or a ratunculus, and like, or even other species, like how would the body maps be similar or different at different levels of organization?
Well, in terms of the size and shape of different brain regions, it's very clear that you can, I mean, I do this with my students. I show them the brain of as we were talking about last time, the brain of a rabbit and a cat, the ratunculus and the catunculus. In other words, the body map on the sensory cortex of the rabbit and cat, and compared to human, and they can pick out, just based on the size, the relative size of the different parts of the body represented on the sensory map, they can tell what the lifestyle of the animal is. And so, in other words, it's the more neurons there are devoted to a particular function, the more precise and the more discriminative, the better spatial resolution, that is, the better detail that the animal detects, or the organism, or we detect. I mean, you know, a clear analogy is the number of pixels in on a screen that you know, the Mac ProBook, or rather the Pro MacBook.
The retina display.
What? Window display. Yeah, the retina display.
Retina.
They boast that the more they have a tremendous an increase in concentration of individual pixels, because that gives much greater detail. You can see much greater detail. So you can imagine if you have if you try to take a photograph on a screen with say a million pixels in the area, each pixel being sensitive to some input, and it's all, let's say, in a 1 square inch area in the back of your camera, that the input is coming into a million pixels. That if you compare that to, say, a camera that has 100 pixels on the screen, you can see that with 1 million pixels, you can get much greater detail in the photograph than if you are dealing with only 100 pixels. It's the same thing on a computer screen. The more pixels there are per unit area, the greater the detail.
Would you consider a cortical column to be a pixel? And like, do you think that they make sense as like a unit of organization?
Yeah, it's true. And the columns are all packed together.
Would you say, so one thing, so, you know, Mountcastle and others like suggested like that there's this common sort of significance of cortical organization based on like a kind of layered columnar architecture. But in terms of like if you look across different cortical areas, there's differences, but they're largely similar
Yeah.
in a lot of ways.
Yeah, because I don't think it's the column per se, it's the number of columns per unit volume of the cortex. It's just, I mean, it's, in other words, a column is, or a neuron is to a column as a pixel is to a voxel.
So is it like, but this broader organization, so like, different types of columns. So like some people would talk about the significance of like a micro column, like roughly like 100 neurons, maybe developmentally, like crawling up the same radioglial cell and maybe having some unit there. So there's the idea that this could like represent like some feature in space, but then other people would focus
it
on
does.
like the
No,
macro
it's not.
column
It's
level.
not as if it does something in space. It does. In the case of the whiskers, in the case of the whisker map in rats that Woolsey did, different, and also Wally Welker, that in a particular column, that I would say a particular column is devoted to a particular whisker.
So these would be whisker barrels as macro columns.
Whisker barrels that each, and what the barrel is what you're talking
But
about.
these are
as a
much
column.
Larger though. These are like 100,000 neurons or something like that, correct?
Okay, but the idea is of the barrel is made up of it's like a column. It's like a fat column.
But
so
they're big though, correct?
But they're big, yeah. But the thing is that that one barrel represents one vibrissa, one whisker, and the different neurons in the column, in the barrel, going down deeper, away from the surface, they code a particular angle of deflection of that whisker.
And so the micro columns would be giving you this like angle of deflection. Their organization of the macro column would give you the overall vector of the whisker. And this is the whisker.
Okay, barrel. But not only the vector of the whisker, but the whisker itself,
the whisker
as opposed to the neighboring whisker, which has a different column.
Would you say, so I'm trying to make sense of like different kinds of columns. So for instance, one analogy or disanalogy would be like between like an ocular dominance column in V1 and a whisker barrel column, or maybe somewhere else. And so
it's the
how
same
would those
idea,
be similar?
Because the orientation, because that column in the visual system, different neurons in the column represent different orientations of the visual input. So it's very similar.
Would you say that this would be, so for instance, let's say we're looking at the somatosensory, or we're looking at like S1 or where the whisker barrels are these just perceptual? Are they both perceptual and motor structures?
Well, probably. Well, I mean,
like, do they have motor component? Like, can they control the whisker to some degree?
I think, I don't know. That's a good question. In the rat, I don't know if it's a motor control, but I mean, in the human homunculus, the motor, you have the sensory homunculus, the map, the body map, where the hands and the feet and the face, and they're all in different places, systematically arranged linearly over the surface, and that's in that, and immediately in front of that is the motor system, which has the same map, and there's
a slight difference. You pointed out a difference that made a difference last week.
A slight difference is the no genitals in the motor map, but the point is that the face is in the back in the postcentral gyrus for the sensory cortex, and the face is right in front of it in the precentral gyrus, and so the maps are almost congruent, the sensory and the motor. So, and
but you're going to have somewhat of a division of labor, though, based on the connectivity, where you have like, so do you have, for instance, in a rat, and this, do they have motor whisker barrels, or would this just be in the sensory side?
I don't know. It's a good question, because I know they Woolsey mapped it according to um, the sensory response. Uh, I don't know. I haven't followed
So
that up,
part of what
but
I'm trying to figure out is so I was very influenced by this book that came out around 2004 or 5 called On Intelligence by Jeff Hawkins. And so he basically tried to argue this idea of like all of cortex having like a common algorithm. And it's just like the different functions are where you are, like the location, location, location. But in more recent work, he's making these arguments that the cortical macro column, that each one is a complex modeling system capable of having both sensory and motor properties everywhere, no matter what. But rather than it being like, he makes a variety of claims I have mixed feelings about at this point, but I haven't ruled them out. But the idea is that, so for instance, like he'll argue that you lay down this basic cortical circuit, like with the hippocampal and entorhinal system for navigation, but that this laid the template for the rest of the cortex, and that every column, he argues, is a system that has like things like general head direction cells, general place cells, general grid cells, and that each one is doing like object modeling. So like if you were talking about something like a whisker barrel, the whisker would be encoded in the macro column as a kind of object with a reference frame for its coordinates, and it would be its own sensory motor object. And the claim is that all of cortex is doing this. The mixed feelings I have is I'm wondering whether you're having different types of columns depending on whether you're actually getting the closure of an action perception cycle. So like, are you like actually having something that like you can wiggle independently, and then you get the feedback from it, and then this is like an object. But like the idea that like all of cortex everywhere is, it's hard to summarize this model. The new book is called The Thousand Brains Theory, and the idea is that he should call it the 100,000 brains theory, because he's saying like for a human, but like every column is doing the same thing of this. And
every column does its computation.
Yes.
It's not just yeah. I mean,
I'm open
it's
to
not.
this, but it's the
I'm
question of
open
what's
to
the
it too. It's
computation.
not a bad idea if it's
Yeah.
true. I'd like to know what the evidence is. I mean, there does seem to be evidence for it in the sense of there in a particular column there seems to be at least sensitivity to different orientation in the whiskers and also in the visual columns that depending on the angle of the line, you get activation of one or different neurons within a particular column, and the column is representing a particular point in the photographic plate. It's like that.
And I sound very skeptical of the idea, but the one idea is like the question is like, what's the nature of the modeling of the information for the column?
Yeah.
So like the idea that like you're actually having an object with its own reference frame for something like you're describing for whisker, I'm like, sure, but is that all of cortex? Is that every ocular dominance column?
I don't know. As I told you, it's relatively recently that I've been brought kicking and screaming into the cortex, because I was happy in the lower brainstem and happily spending my career in the lower brainstem, and suddenly as a result of the research I'm doing, I'm dragged into the cortex, into cortical function. So I have to learn all this stuff. Anyway,
there's other places where I'm curious, like I'm going to drag you there, but who else is dragging you in that direction?
My research, that in order to understand studying orgasm, and there's a lot of activity all over the brain, not just in the brainstem, the cortex, hippocampus, amygdala, basal ganglia, that's all, and the pleasure, trying to deal with the difference between pain and pleasure. It involves the basal ganglia, which that's a nightmare, and interaction between locomotion and motivation, which is, I think that's really a fascinating aspect, because the basal ganglia are doing movement and motivation, and they're intimately linked. And with dopamine the mesolimbic system and what is it, mesolimbic and mesostriatal system,
Nigro, yeah.
nigrostriatal system, so they're intimately linked, and so those two things go together, and uh, well, a rat that's bar pressing, it's expressing the motivation. So the movement and the desire are intimately linked in the most primitive way. But that's another story. But I wanted to get back, if you don't mind. I just wanted to get back to sort of complete the thought that you raised in the beginning of that you can tell something about the life of the animal, of the organism, by looking at the shape of the brain, and the relative size of different regions. So, for example, and the principle is that the more neurons there are devoted to a particular function, the greater the resolution, the greater the precision of the movement, and the greater the resolution of the sensory input. In other words, how detailed, technically it's called the 2-point discrimination threshold for sensory system. In other words, how far do 2 points have to be from each other in order for you to say that they're 2 points, rather than just 1 point, which is an indication of the detail of the detail that the system is capable of. In other words, it's really very, very surprising that, and I use this as a demonstration in my class when we have face-to-face classes, that you take 2 toothpicks and put them, say, an eighth of an inch apart, and you're not looking at your finger. You put these 2 toothpicks an eighth of an inch apart, touch your finger lightly, and you can tell it's 2 points. You take that same thing, the same pair of toothpicks, and touch it against your arm, where we get an injection of a COVID injection. They say it's 1 point. So okay, then you move the points farther apart. You move them an inch apart, and you press it against the same place in the arm. And they say, you say, how would you describe that? And they say, well, I feel a point. You're poking me with a point. And then you put the toothpicks 2 inches apart, and they say, yeah, you're poking me with a point. You have to go more than 2 inches and then they say, oh yeah, there are two different points. That's the idea.
Does this have exactly the same scaling as like a Penfield homunculus, or is it different?
That's exactly that. So what I do, I show a picture of the Penfield homunculus, and a picture of there's a very nice diagram of the 2-point discrimination threshold. In other words, how far apart the 2 points have to be in order that they're perceived as 2 points rather than 1. And on the finger, it's about 4 millimeters, which is about an eighth of an inch. 4 millimeters on the finger, but it's 44 millimeters on the arm. In other words, you have to be 44 millimeters apart, which is like 2 inches or more, or approximately 2 inches. I don't remember exactly the conversion, but they have to be very far apart like that in order for you to detect that it's 2 different points on the arm. And when you look at the homunculus, so there's a map of, it's a picture of a guy lying, a body lying prone with the 2-point discrimination thresholds shown as vertical lines, and it's they're very, very short for the finger and the lips, and very, very long for the upper arm and the shin, the lower leg. And when you look at the map, at the Penfield map, the fingers have a tremendous representation, many, many neurons, Actually, the finger and the hand, the hand and the face make up 50% of the homunculus. That means I have
This is
50% of the sensory neurons in the cortex are devoted to the hands and the face. And the rest of it, the rest of the enormous body has a tiny representation on the homunculus. And that's why when you see a picture of a gnome made up of, a three-dimensional caricature of that, it looks like very grotesque, a giant head, giant hands, and tiny body. And a big tongue also, because that also has a tremendous representation.
okay. So this is raising an issue for me. Mind if I share my screen?
Sure.
So I wrote this paper recently that I published in Entropy. I call it the radically embodied conscious cybernetic Bayesian brain. Decided to make a very long title. And part of what I'm doing is I try to like lay out as many of like my ideas as I could in one place in terms of how I think they go together. And so this would be, in this figure, it's just the idea of cortex of what if you are having like these columns doing some sort of common process, and then you're abstracting patterns of patterns throughout.
Okay. Yeah.
I mapped this onto some ideas from machine learning that I think might be relevant, but that's not the thing to get into here, although we could talk about that later at some point if you're interested. But there's like the idea though of like trying to think of like a hierarchical rhythmic syntax of integration. And that's what this figures. But what place I want to get to is there's, here we go. So the claim was that I make at one point is that the Penfield homunculus at some point gets like re-represented into something that's more topographic. So you were describing yesterday, like you could see in this hierarchy of body maps, like if you had like the magic cap, which would have the perfect spatial and temporal resolution, every neuron, we've tracked every one, you could see these like electrochemical objects potentially at different levels showing up again, like as you're doing the jumping jacks. It would be following through.
Exactly. And it would be, and in order to get that precision up to the cortex, the precision has to be there at every relay stop. So it has to be in the medulla for the nuclei cuneatus and gracilis, and it has to be in the thalamus, the sensory thalamus, the ventral posterior lateral thalamus, and up to the cortex. So you have to have the homunculus at each of those levels.
So here's the question. So in an earlier version of this, I drew this. The idea was like the Penfield homunculus. The hypothesis is it gets re-represented into like a quasi-topographic representation that allows for better coupling across this control hierarchy, maintaining the topography.
I agree completely.
But an interesting study I found suggested, so I was wondering whether this like inferior lateral parietal in a human, I know that might be a little bit shifted in a rodent, but the idea that this structure might be like what lets you have like the lived body, like the body as experienced. And we were discussing before, you were saying you thought it would be like close to the motor system. And then I was wondering, well, how much motor is in their parietal? So maybe there's one in the front, one in the back. Maybe their coupling is what makes them conscious. I'm in some of the models I'm working with, I'm thinking it's the back one, but maybe some people would say it's just the front one, if there is such a structure. But
no, it's probably no.
Go on.
I don't think it's. I think it does have multiple representations. The body maps or some aspect of the body maps must have multiple representations. I'm looking at the picture that you have for the upper rightmost picture, where you have various body maps in various places in the cortex. I mean, another place that we didn't mention before, that I didn't mention before, is the somatosensory 2 area, which also has a body map. And that it looks like you may be drawing that in the middle of the temporal lobe, but it's probably in the insula, because
Oh, which
the
one?
insula,
I mean, this one here, or
no,
or-
that whole brain image. I'm talking
yeah.
about the whole image with look, you have body representations
Oh, so this one here
all
was
over.
the insular one. I'm like showing it like it's tucked in there.
That's right. That's
Yeah.
right. That is a, but that has a different, no, the beauty of it is that they're all, they all have different types of um of response. In other words, in the insula, the somatosensory 2, there are neurons that respond not only to the body map, but they not only, like in the somatosensory cortex, S1, the say it responds to tapping a finger. A small group of neurons responds to tapping a finger, one finger. But in S2, which you have the little guy with the arrow going to the insula, that's where S2 is, and the operculum of the cortex. It's insula and operculum. Then not only does that finger, not only if you record from a neuron in S2, not only does it respond to that finger, the same finger, but it may also respond to the same finger on the opposite side. Or it may respond to a visual input. In other words, the somatosensory 2, that's the idea of integration. Integration means that you may think that it's sort of sloppy, but that's what integration is, because not only the beauty of the somatosensory cortex S1 is that it has very, very specific response, that a particular neuron responds to a very specific part of the body and only that one spot. But in S2, that particular neuron that
Clear
will,
mapping across the pixels between the
what?
like the pixels, the
Well, it's like
columns. Like there's like a clear like specificity
in
of what
in
corresponds to
S1, in
what.
somatosensory
S1.
1, the primary somatosensory
But
cortex.
by the time we
Yeah,
get to S2,
but when you get to S2, not only do you get if not only does a particular neuron in S2 respond to that same finger, but it also responds to other fingers on the other side of the brain, maybe to auditory and tactile and auditory and visual input. It may respond. It's a multimodal neuron, but that means that it's involved in responding to both, say, bilateral input that helps to relate the left and right sides, or if it responds to a visual input, it might be involved in eye-hand coordination. So in other words, it's more the S2 neurons are more multimodal. They respond to multiple modes of input, of sensory input,
so
not just one. And that is more integrative.
above S2, what would you say would be hierarchically higher than that? And how would it change again?
Well, it could S2.
So I guess what I'm wondering is like, is there a level on top? So we start out with like
Yeah,
this homuncular representation of the Penfield homunculus,
right.
and then S2, you're losing some of the clear specificity of the mapping, but you're getting this multimodal richness.
Exactly.
But I'm wondering, like, is there a
It
level
keeps going.
later
Yeah,
where
I
you
know what you mean.
actually re-represent into more of a
Re-represent.
geometric frame, where actually,
Right.
like, if you trace out the connections, the location,
Yeah.
location gives you basically something that's a little bit actually pretty homuncular, where
Well,
actually
okay.
left is left and right is right to some degree in the connectomics. If you trace it out.
I know what you mean. So what I would expect possibly is that it would project to some place that also where neurons respond also to proprioception. So and there would be a body representation of the proprioception, because that in other words, to relate the sensation to the movement, there's got to be um, that's a different level of integration. And I don't know where. Well, I know
So I'm
proprioception.
thinking lateral parietal cortex.
It could be. It could be.
And
Yeah.
possibly a little inferior. I'm not sure in the human, but the
Well, it
thing was
looks.
so to loop around a thread before I forget. So one thing that's interesting I found though, if you look at like the body representations of, or if you look at like of that area, it seems that actually the proportions of the parietal body maps, I forget if it's inferior, it might be superior lateral parietal, but they're not of the Penfield homunculus.
Right. Exactly.
But that's interesting because like I was wondering, okay, so maybe it's actually like the Penfield homunculus, a lot of this would be very fast operation. So it's like you actually don't want to have like, you need exquisite sensitivity and control of like your
Yeah.
face,
Yeah.
but you're not controlling every single thing of your face. That would be crazy. And that's
Right.
like not what it's like either. Like you feel your face, but it's very like a lot's going on, like reflexes of the face.
Well, yeah. I mean, think of the eye blink reflex. That's
I'm not
controlled by
controlling that.
What?
I'm not controlling that.
Right. But and we're blinking. So I mean, there's something going on that the blink is responding to, say, trigeminal input from the cornea, and you blink when the cornea when it gets irritated, you blink, and so it's a response. It's the eyelids are controlled by the facial nerve, and the cornea sensation by the trigeminal. So there's, without us voluntarily thinking about it, we're activating that pathway from trigeminal to facial to control eye blinking in response to the eyes getting dry, and so there's got to be integration of that body map somewhere. And I mean, you drew. It looks like if you could move that last picture again, I really like that diagram. I'm going to use that. The Because it looks like you have. I mean, is that the eye on the hand in the occipital lobe,
Yes.
and the eye in the hand, and the motor, the eye field?
So this would be like roughly like maybe like DLPFC here, and then this would be like um, inferior or rather lateral parietal.
Okay,
Or
lateral parietal.
posterior parietal, but like some combination of part of the claim. And I guess in a rodent would be different because it's like snout eye coordination, but in a human that you're getting this like intense intermingling of hand representations and eye representations,
right? So
on
that's
the motor
that.
and perceptual side.
Okay, so that is an example of integration where one neuron responds to two different inputs, hand and eye, and that is most likely going to be related to eye-hand coordination. And the advantage of having a neuron that responds to both is that it means that there's an intimate connection between the eye and the hand, which is true. So it's another kind of homunculus. Yeah, I completely agree. So it's but maybe a nice way to express it. I don't know how you express it, but for me, I would say that it's a conceptual homunculus,
Mhm.
because if there are neurons that respond to both eye and hand input, that's got to be a higher level than the kind of slave-like response in the somatosensory cortex. So, and the brain is organized that way. I mean, they're every, the different functions are represented by different neurons. There are some neurons that are slaves to the input and slaves to the output, with that, and slave meaning that they have
In the
one
technical sense,
in a
like
technical sense.
directional entrainment.
Yeah, and they respond slavishly to the touching of the thumb. That's their job.
Hmm.
It's a slave in the sense of slavish, meaning they don't have any option. They just do it.
Or in the cybernetic sense of like,
In the cybernetic sense, okay.
you know,
But then when you get into something like a neuron that responds to visual and hand input, that is more conceptual because it's involved in coordination between the touch of the finger and the movement of the finger, or the touch of the finger and the position of the eye. So it's that kind of thing. So it's more um, integrative or more conceptual, higher order processing.
at one point in this paper, I make something of like the etymology of conceptual as like to grasp with. And the claim that I try to pursue is that every thought on like, even like in an abstract conceptual level, to be experienced ultimately, and to be controlled, ultimately is cashed out in some sort of sensory motor perception action hierarchy. And that actually ultimately you have to ground it in some sort of intermediate level physical representation of some kind. Like even something like where like abstract like justice, it would be like maybe a montage of images or a feeling in the body of rightness, but that actually the concept of justice, or like maybe there is some sort of like abstraction of like relations that's, I don't know how you would do it, like I don't know, weighting scales or something, but that ultimately though there's some image, either
Right.
somatosensory or visuospatial.
Yeah. I think this is my crazy idea, but
I love crazy ideas.
I think the highest, I think the place where consciousness resides is in just before the motor output,
At the homuncular level.
at the homuncular level, at some conceptual or that there must be, in other words, the reason for that is that there's integration between, at some point, just before we do something voluntarily, I mean, quote voluntarily, we make some voluntary action like picking up a cup, that the everything, the whole system has to funnel down into all the slavish inputs and all the slavish outputs, but all the slavish inputs and all the integration of all the senses and all the motivations and all the factors and memory and everything, that all has to funnel down into a decision or into a movement based on a decision. So the high, it seems to me
You
that
have to ultimately interface with the spine and the full hierarchy of homunculi that are actually directly connected to the spinal motor pools.
the spinal
Ultimately,
cord,
you have to interface with that topographical system to actually generate the behavior.
Exactly. So right at the point that the behavioral movement is made, that is the high, just before the actual motor output, there, that is the highest form of integration, because that's where the decision is. Everything is integrated, everything comes together to do a simple output. And because it has to be simple, because we can't do trillions of computations, our awareness has to be simple at some level.
You need to reduce the dimensions.
It has to be, well,
you need to be
has
tracking the school of fish rather than the individual fish.
That's
like,
right.
Like the overall trajectory.
And the easiest way to track the whole school of fish is by responding to the water current that they generate. That's a simplification of
So
it.
So I've been wondering, like from earlier in the thread, I've been wondering whether, like lateral parietal, maybe inferior lateral parietal, is one of the primary places where you get that. But then you mentioned the 2-point discrimination. And so I'm having trouble here, 1 second. Okay. The 2-point discrimination, you were mentioning that it's of a similar scale to the homunculus of Penfield's homunculus.
Exactly.
But in this one study though, I found, it came out, I think in 2020, it was looking at what I thought might be this representation that's now re-represented in a way that allows for this interfacing with the spinal homuncular hierarchy. They are saying it's not of the same proportion, and that actually it's a normal, it's like big arms, but it seems to be more in the proportion of conscious controllability. And so it's like,
yeah, well,
there's
that's
a head,
conscious
but it's
control.
not a giant head.
What with conscious controllability
Big arms,
means
like
higher,
this,
of greater
you know.
precision.
But here's the thing, though. You though were mentioning though that, but now I'm thinking of 2-point discrimination. That seems pretty conscious to me. And so I'm wondering why there's this mismatch of, like, if you look at the M1 or the S1M1 Penfield homunculus, it's on the scale of conscious resolution of this discrimination. But then if you look at this hierarchical higher one, which I'm wondering might be like the neural and computational substrate of the lived body, or one of the key places, it's not showing that same resolution. And that's the puzzle I'm having based on what you just said. Like, why did you lose that resolution in this at that level of control?
Because, okay, here's the beauty part. You
I'm ready.
lose the resolution because you have integrated all the slavish input resolutions, you filtered it down to simplicity, and it's the simplicity that is required for making the movement. You can't be trying to figure out all the trillion computations simultaneously if you want to pick up the cup. It has to be simple. So maybe the highest, it sounds paradoxical, but maybe it's not paradoxical. It's not paradoxical because in order to make a final output, it's far simpler than trillions of activities. So in the paradoxical sense, the highest level of integration is where everything is filtering down to activating a relatively few neurons, and that's where I think consciousness is.
Okay, I think you just
consciousness has to be simple.
I think you just resolved the confusion for me. So it's like, and that's in this figure, that's part of these, like, I was thinking of them in like machine learning terms of these kinds of but this idea of like these filters, this funneling and dimensionality reduction. That's what I was thinking in terms of these cones.
I like that dimensionality reduction. That's beautiful. I like
or compression, but the
well,
idea
it's not compression, it's integration.
Yeah, you're right. It's not just compression. It's actually synergistic. It's
integrate. It's decision.
So so while in theory, I would know if you put like those toothpicks on my face, or just like in theory with my hand, like you do the toothpicks, I would have that resolution. However, under normal circumstances, I go to grab the cup, I'm hammering the nail, I'm not paying attention to that fineness. I'm actually, under normal circumstances, I'm taking advantage of the fact that I've had this funneling, this filtering, this integration that is able to coarse grain over those details. I'm taking advantage of that for the control.
Yeah,
if I wanted it, I could feel like a little rough piece on the hammer, but that's not what I'm doing when I'm hammering. That's gonna be bad hammering.
That's right. However, if you go to pick up a cup and there's a broken shard on the cup, and as you grab the handle, you get poked in the finger. You know exactly where you know which finger to pull away from that particular region. So that's the beauty of the complexity of our brain, that we can walk and chew gum at the same time. You can
If I
do,
need it, that information's there to tell me
if
exactly
it
the.
became salient, you would respond to it.
Mhm.
But yeah, it's true. We do, as a matter of fact, I can't remember if I mentioned this to you about that that we as you say, you when you pick up the cup, or when you're using the hammer, you're not thinking about a rough spot on the handle, but um, because we focus on one thing, and we do ballistic movements, and that was what that magician, when he gave the lecture at the Society for Neuroscience presidential address, he had a magician. Did I mention that before? There was
So,
this magician, and
yeah.
he was saying how we don't pay attention when we do something like reach for something, or have some uh, activity. We have the initiation. We are aware of the initiation. We're aware of the termination, but we're not aware of uh, the distance of the ballistic. It's a ballistic movement, so we're not aware of every step along the way. We're only aware of the beginning and the end. And he said, and that's what pickpockets use, that because they they take advantage of the fact that we're not watching during that ballistic movement, because we know we see what starts, we know what to expect as the end, but we don't pay attention to what's going on in the middle. And that's when he lifted the wallet and a calendar from the president's jacket, without in front of 1,000 people.
But necessarily we want this, so in that case we don't, but we want this normally because that's what lets us pick up the higher level harmonics.
That's right. And so
that's
we
what gives
weren't
us the control.
so when the president came over to him uh and introduced him, and he reached over to and put his hand on the president's shoulder, that's when we knew what was going to, we could tell the intention. And he was going to put his arm out to put on the shoulder of the president, which he did. And it was at that moment that the magician lifted his wallet with another hand, and in front of 1,000 people, and nobody saw it. And then he gave the wallet back to the president after he talked about how we don't pay attention to the ballistic part of the movement.
Something I wonder about is how many ways do we have to, like you're mentioning earlier, like the mesolimbic dopamine, the nigral motor
Striatal.
dopamine, nigrostriatal, but I was wondering, like, how many ways are we being conditioned with respect to the implicit motor functions of our thought without realizing it? So, like, for instance, how many
All our
magic tricks do
lives.
we pull on ourselves and
All our
without
lives.
realizing, because we want to, like,
That's how we develop skills, because we have to develop these ballistic movements.
but how often are we, like for instance, trying to, like, among the skills, pulling magic tricks on ourselves if they, like, help us to, let's say, like, hide from an uncomfortable truth or something like that,
Sure.
like
Yeah,
what
I'm
extent
sure
do we
it's
do that?
yeah, I'm sure it's true.
I know I do it.
Yeah. So
Confession.
so if I could just get back to the
Sure.
same point, we could get these interesting sidetracks. But so the homunculus I show the homunculus of a rabbit and a cat, and the hands, the paws in cats are gigantic. They're like the hands in humans, and the paws in the rabbit, the representation of this in the homunculus, they're tiny. But so so there's a tremendous difference in the paw representation in rabbits versus cats. And of course the students say it's because the cats are predators,
But
and
in
the rabbits just run.
theory,
What?
in theory, there might be a something of a difference. And, like, let's say there's like an additional, like, posterior parietal catunculus. The paws might have, like, a slightly different thing going on. I'm
Sure.
wondering. Like I'm
Yeah,
wondering, like,
yeah, because control, yeah,
yeah,
grabbing as opposed to running. The rabbits don't grab, cats grab. So that's represented. Not only is
maybe
the
focus
map,
particularly on the claws.
yeah,
Maybe
yeah, it
it's
is.
like
Yeah, it's an
that's the
individual,
point.
the individual claw.
That's where you'd want the real exquisite control, is like
That's right.
which claw is digging into which amount.
And the control of the nails of the claw.
So the pad might kind of go a little bit smaller, but
Yeah,
then
exactly.
it's like
That's exactly right. Yeah. So not only is that
the
true,
lived body of the cat.
but there's the size of different parts of the brain. Like for example, I see I show them a picture of a shark brain and a frog brain, and look at the cerebellum, and the frog has a tiny cerebellum relative to the rest of the brain, whereas the shark has a gigantic cerebellum that covers the cortex. It goes over the cortex. It's so gigantic. And I say to the students, what can you predict about the lifestyle of those two animals, knowing that the cerebellum controls motor coordination? And they say, well, the frog sits on a lily pad, doesn't do anything, it just flicks its tongue out
Just
to get
using
an
the
insect.
tectum to like the caliculi. Just
The caliculi, but
it does all you need.
but the cerebellum is tiny. But in the shark lives in a three-dimensional world, going up and down, and
The shark is like a submarine. It's
like a
it
submarine,
needs to be
and
an attack. Yeah.
and chasing prey with his whole body, the whole thing.
The frog
And
sits and waits usually.
that's
And
right.
just the
Frog sits
colliculus
and wait.
the
It doesn't have
colliculus
to.
salience map is enough.
That's
But
right.
if you're a submarine going in battle with other submarines,
You have to have the dogfight.
yeah,
Yeah.
aquatic dogfighting.
Or chase predator, or chase the prey if you're hungry. You gotta. So that's one example. Another example is a beautiful example is two species of buzzard. There's a carrion buzzard and a predator buzzard, and they're the same species, but and there was this beautiful demonstration shown. A guy, he was studying these two different strains of the species, and and what he did, he put a carrion, a dead animal, under a stovepipe, a tall stovepipe, maybe 20 feet high, with a fan blowing air up the stovepipe, and then he observed that the carrion buzzards flying overhead would come down the stovepipe. They would fly down into the stovepipe, but the turkey buzzard would ignore it, just fly right past it. And looking at the olfactory bulb of the turkey buzzard versus the carrion buzzard, the carrion buzzard had an enormous olfactory bulb relative to the rest of the brain, and the turkey buzzard that goes after turkeys, it's a predator, had a tiny olfactory bulb.
So that it's enslaved to the olfaction.
That's right. Well, it enables olfactory sensitivity as opposed to not necessary.
And to a large degree, though, like the strongest shape around behavior, the strongest shape around the
that's right,
the life world of that
because
creature.
the more neurons there are, the more flexible that behavior can be, the more responsive to different differential stimuli and differential movements, all that. So it's and birds are notorious for having tiny olfactory bulbs, almost invisible. They're tiny, but here's this carrion buzzard that has a gigantic olfactory bulbs. Another thing is you mentioned the tectum. In the case of birds, the tectum, the optic tectum, the superior caliculi, they become so enormous in birds because they're involved in visual visual
like
integration
owl,
in space.
Eagle or a hawk. You'd expect them to have like particularly large.
Not only is it large, but it becomes a major lobe of the brain. if you look at the brain of a sparrow, for example, the optic lobe is enormous. It's bigger than the cortex. It becomes its own brain structure, not
Also
just
useful for avoiding, even if you're not a hunter, just avoiding collisions.
avoiding collision. Not, I mean, I marvel at sparrows. You know, I see a chain link fence with, you know, with
Going in and out, not
little
hitting.
diamonds, and the sparrows, they fly right into it and land, you know, they land and stop right in one of those little diamond shapes, spaces that's barely big enough for its body. So to be able to get that precise aim going at high speed right into the fence and stopping on a dime and not and folding the wings so that they can fit in that diamond, which is practically the same size as the bird itself. I mean, it's remarkable.
That might be the strongest determiner. Like, it might not even be the case that like more visual hunters have even proportionally larger like tectums, because maybe it's like it's just a bigger factor, just that sort of, if you're making fast decisions based on the integration of the visual and your appropriate juxtapose, like you need that takes you to
precision. You need the precision.
Hey,
yeah. How we
gotta run.
Oh, okay. All right. So in other words, not only is it more neurons give you better precision and more sensitivity, more precision of sensation and movement, but the greater neurons, it can be so much greater that it actually becomes a shape of the nervous system. And that's true in the
visible
lumbar,
to the naked eye.
Visible to the naked eye, easily visible to the naked eye. The lumbar and cervical and lumbar enlargements.
You don't even need the magic hat to see this one. You can just
not at all. You just open it up and look at it, and you can tell. I mean, by looking at the spine. This is another example. Looking at the spinal cord of a reptile, or I mean, of an alligator versus a turtle. There's a tremendous difference in the spinal cord. We can talk about that next time. Or I can show you the picture of it. It's just by looking at it, you can tell the difference between which one belongs to the alligator and which one belongs to the turtle.
Okay, I'd like that. So let's dig into the spine next time.
Okay, sure. The spinal cord.
The spinal cord.
From the spine. Yeah, that's a source of confusion. But okay, yeah. Sure. I have a nice story about the spinal cord. I love the spinal cord. Yeah.
Well, yeah, I'm so you've been dragged into the cortex. I want to be dragged back into the spine.
Oh, sure. So, the spinal cord.
Spinal cord. Thank you. I'll talk to you soon, Barry.
Okay, great. As always, Adam, this is really. Can you send me your paper? That paper, I'd like to use that. I'd like to use that figure. It's really, I love it. It's a great figure because, yeah, there are all kinds of. Not only are there homunculi all over the brain, but there are different kinds of homunculi, and that's what I wrote in my theoretical paper on consciousness. So anyway,
and we're going to dig into that some more too.
Well, we could do that, and also the idea that the whole filtering, the whole thing of funneling down and filtering into simplicity, that I think has to be where consciousness resides, our subjective awareness, because it has to be simple. We'd be hopelessly confused if we have to pay attention to a trillion computations. It has to be. It has to come down to nuts and bolts.
So and we can make it easy. Coming down to a kind of action-oriented body map.
Exactly.
Maybe couples with a visual spatial field.
That's where consciousness is.
And maybe it is just an action-oriented body map coupling with a visual spatial field.
The action-oriented body map, just before the action, it's the action-oriented body map that is where our consciousness is.
Space with the lived body at the center, where the body
now we just have to figure out
abstraction for control
where that is. But it may be all over the brain. But it's conceptually, it's got to be somewhere, and it's got to be simple. A baby has to use it.
Let's talk about that some more next time too.
Okay, a baby has to be able to use its brain. So it's got to be simple. Nobody's teaching it. Animals have to be able to use their brain. They're not paying attention to the trillions of computations. Okay, that's okay. We're done.
Could be inferior lateral parietal. We'll see.
Okay,
Talk
whatever.
to you soon.
Okay, have a
Bye.
good week.